not getting. Okay, then I will start. A very good afternoon to everyone and welcome to this 23rd webinar, Naidaniki 24, organized by Rognidan and Vikruti Vigyan PG Association. This webinar is approved by MCIM Mumbai, myself, Dr. Deepali Amle, HOD and Professor, Rognidan and Vikruti Vigyan Department, CSMSS Ayurved College, Aurangabad. As a coordinator, once again welcome you all. First of all, I would like to thank Dr. Subhash Wage sir, President and Organizing Secretary, RVPGA, for organizing this Naidaniki webinar since last 2 years. He has started this webinar series in 2021. Sir has given me this opportunity to coordinate this Naidaniki webinar organized in January 2024. I would also like to thank the esteemed members of RVPGA, Dr. Avinash Ralke sir, Dr. Santosh Chavan sir, Dr. Vaishali Ganorkar ma'am. Today's webinar is on new techniques in microbiological diagnosis. And our guest speaker, Dr. Yadnesh Thakar sir, will explore different aspects of microbiological diagnosis by discovering the world of microorganisms for our PGs. For this webinar, our chairperson is Dr. Abhinandan Mukhe sir. He is principal, professor and HOD Rognidan Department, Bharati Vidyapith, Ayurved College, Kolhapur. Before this, we have to make a tour around our RVPGA at a glance. So, I request Dr. NMB Shia ma'am, Assistant Professor, Rognidan and Vikruti Vigyan Department, Parul University, to introduce our RVPGA Association. Towards you, ma'am. Thank you.
Thank you. Thank you, ma'am. Good afternoon and namaskar to respected Professor Dr. Subhash Wage sir, Naidaniki Organizing Committee members, Session Coordinator Professor Dr. Deepali Amale ma'am, Guest Speaker Dr. Yagnesh Thakkar sir, all the respected faculty members and beloved students. I am Dr. Anambishya, a proud member of RVPGA. I am here to speak a few words about our organization RVPGA and its significance. Our organization RVPGA was formed in the year 2016 under the able and dynamic leadership of Dr. Subhash Wage sir, with the aim to give the legal status of pathologist and radiologist to the PGs of Rognidan. Our organization is duly registered under the Societies Act of 1860 at Charity Commissioner Office, Nagpur. Postgraduate students, teachers of Rognidan and Vigyan can become our member. Ours is the only organization which is fighting for the lawful rights of Rognidan PGs. Even the NCSM had also directed State of Maharashtra to consider RVPGA as a stakeholder in the constitutional pathology regulatory body of the state. RVPGA is pursuing this matter with state and central government. Not only this, RVPGA is constantly pursuing the implementation of NCSM salary structure in all the private Ayurvedic colleges across the nation. All the communications are uploaded in our website www.rvpga.com. Our activities include regular conduction of online webinars and offline seminars, both on national and international platforms. So far, we conducted 23 national webinars on different diagnostic topics and a national conference on X-ray diagnosis at Nagpur. Hands-on training program for PGs at our partner NABL accredited pathology laboratory at Mumbai, Nagpur and Pune. We have planned a microbiological hands-on training program for our PGs on this 9th March 2024 at Nagpur. Publication of research papers of Nidan students and faculties in our peer-reviewed journal, International Journal of Diagnostics and Research, IJDR. Interested can visit journal site www.ijdrindia.com. Our journal is enlisted in 8 databases. 2 issues have been published so far. Edited book volume series through our international publication RIPI, which is registered in Government of India's ISBN portal. Those who want to contribute book chapter can visit RIPA website www.rvpgaiip.com. We have published All India Rognithan PG thesis book consisting of all the Rognithan PG titles from 1985 to 2023 for the easy reference to our PG students and faculties. We do legal fight for the issues of Rognithan and Vikriti Vigyan PGs at appropriate forums. We have our YouTube channel and Facebook page where you can see our educational activities. Thank you, ma'am.
Thank you, ma'am. Thank you for your valuable words. Now, I will introduce our today's eminent guest speakers, Dr. Yadnesh Thakar, sir. Sir is Director of Vishakha Clinical Microbiology Laboratory. With the wealth of expertise in the field of microbiology, sir held the post of Consultant Microbiologist at Care Hospital, Nagpur. He is meritorious student of Government Medical College, Nagpur in both MBBS and MD Microbiology. Formerly, he held the post of Associate Professor at Government Medical College, Nagpur. His notable contributions includes 37 research articles, co-authored 3 manuals on clinical microbiology and immunology, authored a manual on hospital infection control methods. Sir is honored by Dr. Pushpa Jagtap Oration Award and Dr. C.N. Chowdhury Teachers Award. So, without further ado, let's welcome our guest speaker, Dr. Yadnesh Thakar, sir. He will share his insights and knowledge on new techniques in microbiological diagnosis. Towards you, sir. Thank you.
Thank you very much, madam, for a very nice introduction. I am sharing the screen now. Now, can you all see my screen?
Yes, sir. Yes.
Yeah. So we can proceed. Respected chairpersons, then President Dr. Subhash Wage sir, Dipali madam, Ambitia madam, and distinguished faculty of Vikruti Shastra in Ayurveda. It is really heartening to see that you people have decided to organize something related to microbiology, because during last about 22 decades, microbiology has become one of the most important branches in the medical fraternity. And with the advent of COVID, people have started understanding also that microbiologists, they do a lot of work in the laboratories. Now, microbiology has been a part of pathology. And in our field, in medical colleges, the microbiology was separated from pathology at around 1973-74. And since then, it has been a separate branch. And you will wonder how much microbiology has grown over these years. Now, today, we will just have a bird's eye view of what exactly is happening in the microbiology laboratories. Now, you can see here that I was told to talk about the emerging technologies in medical microbiology. I thought that it should be from point of care testing to the next generation sequencing. So we will try to cover everything. But if we go into the details of each and everything, you will see and you will find that each technique, it requires a separate session if we have to go into the details of all these things. So today we will discuss only the bird's eye view of what exactly we do in microbiology and how automation has changed the scenario in the diagnostic microbiology laboratory, and now how the things and why the things have been made in such a manner during the so many years. So let us start without wasting much time, because I have some 50 odd slides. So let us go ahead. Now, somebody wanted to ask.
Dr. Hemant, can you address their issue? Were they?
Okay, sir. Okay.
Okay. I think the slide
Continue,
is...
please.
We
will sort out.
Should I continue?
Yes, sir. Sure.
Yeah. See, if we know that we all know rather that these infections are caused by 4 kinds of microorganisms: bacteria, fungi, parasites, and the viruses. And these the commonest infections are caused by the viruses, followed by bacteria, whereas parasitic infections, they occur, but they are not that common as compared to the viral infections and bacterial infections. And fungal infections are usually the skin infections. And if we get the disseminated infection, we get it in the immunocompromised individuals, or the individuals in whom the immunity is little low. Now, if we look at the diagnosis in microbiology, in general, if we look at the diagnostic modalities which are available to us, we divide them into the two methods, direct methods and the indirect methods. Direct methods means where we can directly demonstrate the presence of the pathogen. And we know it very well that they are microscopy and the culture. We do them routinely. Every laboratory does these techniques in day-to-day life. And then if we are unable to diagnose by microscopy or culture, we resort to the indirect methods, that is the serology. We also call them as the serological method, that is the detection of antigen or the detection of antibodies. So these are the routine methods which are used in most of the laboratories. And now, since around 2000, the newer methods have come in vogue in the diagnostic laboratories. Previously, this technology was only used in the institutions and for the epidemiological and the research purposes, but now it has come in the diagnostic laboratories, and it has become one of the most important diagnostic tool nowadays, especially for the viruses and the bacteria which are fastidious to grow. And we call them as newer method, or we call them as the molecular methods. Now, in a traditional bacteriology lab, we get the sample. Now, one important thing that people have to remember is that the sample has to be the representative of infection. So, if you have got the infection in the lungs, the specimen should be from the lung. Either it should be sputum or bronchoalveolar lavage or some biopsy or tissue like that. Urine will not suffice if you have an infection in lung. You cannot taste urine. So sample has to be representative of infection. And then when the sample comes to the laboratory, we do either direct microscopy or the culture. In direct microscopy, the commonest thing we do is the gram stain. Then we do the Zn stain or Ziehl-Neelsen stain for acid-fast bacilli and some other special staining techniques. Then for culture, in culture, we actually try to grow the bacteria. We can grow bacteria in culture. We can grow fungus in culture. And we can also grow some viruses, but in the spatial medium, because they will need the cells. So either we grow them in the embryonated eggs or cell lines. So it is beyond the purview of the local diagnostic laboratories. Now, as far as the culture is concerned, the commonest culture that we do is the bacteriological culture. And most of the laboratories are doing hundreds of cultures per day. Now, what we do, we inoculate the specimen on culture plates, incubate them overnight or for 48 hours, and then we see the growth. After we get the growth, we confirm the growth by doing the staining, colony morphology, biochemical test, and then we identify the organism. And once the organism is identified, that the organism that we have grown is the pathogenic organism, then we do antimicrobial susceptibility testing. Now, why this antimicrobial susceptibility testing is done? Because we have got wonderful antibiotics which can kill the bacteria and which will remove the infection from the human bodies. But these bacteria, they are also very smart and they develop resistance to these antibiotics. So when penicillin was discovered, within 1 month, the staphylococci, they develop resistance against penicillin by producing an enzyme which is called as a beta-lactamase enzyme. This beta-lactamase enzyme, it used to destroy the beta-lactam ring of the penicillin, which was very effective in attaching the penicillin antibiotic to the bacterial cell wall. So the antibiotic become useless. So that's how the organisms, they become smarter. And nowadays, we have got hundreds of antibiotics, and these bacteria, they have got hundreds of mechanisms by which they can destroy these antibiotics and become resistant to these antibiotics. You have to remember that if the organism is sensitive to the antibiotic, it means it will be killed by that antibiotic. If the organism is resistant to an antibiotic, it will not be killed by that antibiotic. And nowadays, we find that there are certain organisms which are pan-resistant organisms. Pan-resistance means they are resistant to all the antibiotics which are available to us. And the cost of antibiotics, or there are organisms which are sensitive only to 1 or 2 or 3 antibiotics. They are against rest of the antibiotics, they are all resistant. Now what happens that you are giving an antibiotic to a patient, and these antibiotics are very expensive. So you are spending thousands of rupees for antibiotics, and after 5 days you get to know that this antibiotic is not working. So instead, if you on day 1 itself, if you do the culture, on 3rd day you get the report that to which antibiotics the organism is sensitive. So if it is susceptible to the organism, to the antibiotic which you are already giving, then you did not change the antibiotic. Otherwise, you can change the antibiotic within 48 hours, and that saves a lot of money and the morbidity to the patient as well. And of course, in some cases, mortality also spared. So we do the antimicrobial susceptibility testing and give the final report. And this final report is again, it is interpreted by the clinician, and on the basis of this report, the antibiotic is continued or it is changed. So this is the basic structure, this basic flow chart of the activities that we have in a microbiology laboratory. Now, there are some shortcomings of this routine methods of diagnosis. What is the problem with microscopy? It has got low sensitivity. See, for an ordinary laboratory worker, if you have 10,000 or more bacteria per ml of specimen, then you will be able to see them in the microscopy. But your sample can contain as low as 10 to the power 2 or 10 to the power 3 number of organisms, 100 to 1000 microorganisms per ml. In such cases, microscopy is not very useful. And in microscopy, identification of the organism is not, it may not always be possible, because many organisms, they have got similar morphology on microscopy. Then as far as culture is concerned, it is definitely time consuming, because if you are doing the culture, the organisms will grow in about 24 hours, and in next 24 hours, you will be able to do the antimicrobial susceptibility testing. And then it has got a low sensitivity. And again, problem is that all the microbes cannot be grown on culture media. Like there are some fastidious bacteria, the bacteria which require very special nutritional requirements. So in routine culture media, we cannot grow these bacteria. And again, viruses and parasites cannot be cultured on the ordinary medium which are routinely used in the diagnostic or the clinical microbiology laboratories. Now, as far as serology is concerned, it can give you false positive as well as false negative results. And many times when you are detecting, especially when you are detecting the antibodies, it may not differentiate between the present and the past infections. And therefore, what is happening that the laboratories are, there is an, I think somebody needs to be muted. Now what happens that there is a demand that you give us the report earlier. So they want the shortening of the time and also improving the results. Now, so what is the focus today? Speed. They want the speed. They want the accuracy. And at the same time, they want our test that they should not be very expensive for the patients. So that is the need of the day. And now for that, there is a tsunami of automation in clinical microbiology diagnostic laboratories, and there are huge number of automated systems that are available. Now let us quickly see where automation is available. Automation will increase the speed, it will reduce the mistakes, and it will give you more accurate or error-free reporting. So all sorts of automation, they are available in the microbiology laboratories. There are automated staining machines that are available. Again, there are automated plate inoculators also. So if you have to put the culture, you just put the samples in the machine will inoculate them one by one on the plates. Then there are automated blood culture systems. And after you get the growth, you can identify the microorganisms by automation. Instead of doing manual biochemical test, you can identify them in the machine. So it is faster, error-free, and the better identification is there. And also automated antimicrobial susceptibility testing methods are available. So these different machines are available, and the most common machines that are used in all the laboratories are automated blood culture systems, automated identification systems, and automated antimicrobial susceptibility testing system. These are some of the gadgets that are used in most of the laboratories. Now, they are also based on the conventional techniques. Only difference is that instead of human being doing the work, the machines are doing the work. So that is what is available as on date. Unfortunately, what happens, the cost of these machines is very high. This machine identification and antimicrobial susceptibility testing machine, this machine will cost you somewhere around 12 to 15 lakh rupees. Now, this blood culture system will also cost you somewhere between 12 to 15 lakh rupees for a smaller versions. Now, these are very expensive, and at the same time, their reagents are also a little bit expensive. So the cost per test, it increases. So again, we have to strike the balance between the manual exercise and the automation. So we decide where are we use the automation, and we decide where we use the manual things. So we make the use of the both the things. Now here you can see that there is something called as MALDI-TOF. This is a wonderful machine that has come in vogue. It identifies, after culture, of course, it identifies any organism in about 30 to 45 minutes. So we don't have to wait for overnight incubation for identification of the bacteria. So the machine is called as MALDI-TOF. It means that it is matrix assisted laser desorption ionization time of flight mass spectrometry. So it is basically based on mass spectrometry. Now what this machine does, it does the microbial identification and characterization. It is very rapid, very accurate, and interestingly, the test is very cost effective. So for identification, you require only 15 rupees to 50 rupees for identification of one bacteria. Of course, please understand that direct identification from the specimen is not possible. It is identification after you get the growth in the cultures. Now, there are two systems which are available in the world. One is Vitek MS or Vitek MS Prime, and second is Maldi Biotype. So, these two companies, they are manufacturing this type of machine. Now, although this test is very cost effective, individual test is very cost effective. You can do hundreds of tests at a time in a machine. So if you got a 1 or 2 or 3 samples, again, that is not very effective way to utilize this machine. And secondly, the cost of this machine is very, very high. It has a forbidden cost for the smaller laboratories or the clinical diagnostic laboratories. This machine is very useful for the huge and very big institutions where you get 100, 200, 300 cultures positive per day. Now, the cost of this machine is around 2 crore rupees. So, this machine is not very useful for a smaller laboratories, but we have this machine in Nagpur. One machine is at veterinary college, and another machine is now installed in AIMS Nagpur, All India Institute of Medical Sciences. So basically, this technique is a non-fragmenting or soft ionizing technique. What we do, analyte, that means we take the growth, it is embedded in the acid matrix material on a metal plate, and then nitrogen laser excitation is used to catalyze the charge transfer from matrix to the growth for the desorption. The ions then are separated from that organism, and their mass by charge ratio is analyzed. And mass analyzer is used for detection or creation of the spectral profile. In short, in very simple words, I'll tell you that the protein profile of that organism, microorganism, is mapped out, and on the basis of that protein profile, the organisms are identified. So what is MALDI-TOF? What does it do? It is basically the proteomic fingerprinting of the microorganism, right? And it is very sensitive and specific technique because confidential interval is more than 95% of the all identified microorganisms, especially the pathogenic organisms. Now, what are the advantages of this machine? It has got a high throughput and short turnaround time. I have already told you that in 15 to 30 minutes, you can identify the organism. Now, the question is, why do we have to identify the organisms? See, there are many organisms which are just commensals on our body. There are many organisms which are only opportunistic pathogens. So, if we identify the opportunistic pathogens in human body, then we have to think that if the patient is immunocompromised, then we have to take into account. Otherwise, we have to see that, and we can say that these organisms are just commensal flora, right? And then there are truly pathogenic organisms. So we should know all these things. Secondly, there are some organisms which are naturally resistant to certain antibiotics. So when we use those antibiotics against these organisms, they will be of no use. Say, for example, Pseudomonas, which is a very commonly isolated organism in the hospital. If we isolate Pseudomonas in the OPD patient, we think twice. कि contamination तो नहीं है. But if we isolate them from the hospital, in the patient in the ICU, we definitely would like to treat that patient. And in such cases, if you know that it is Pseudomonas, so you should not give certain antibiotics like the basic penicillins or ampicillin, amoxicillin, or first or second generation cephalosporins, because it is naturally resistant to these antibiotics. Likewise, there are many other organisms which are naturally resistant to certain antibiotics. We have got the list, and accordingly, we have to change the antibiotic in such patients. Now, another interesting part of MALDI-TOF is that it can identify more than 1500 species of bacteria. So it is a huge bank of identification. Thirdly, a very tiny amount of bacterial growth is required, which is not at all affected by the culture media or the incubation conditions. And again, cost is an important factor in our country, so there is a minimal cost per test. But every technology has got its advantages as well as disadvantages. So if we talk about the MALDI-TOF, there are some issues with MALDI-TOF. What you need is a pure culture and not the specimen. In mixed culture, you will have to pick up the most dominant organism. And by mistake, if you pick up the two colonies together, this MALDI-TOF, again, it will get confused or it may give you the wrong diagnosis. Then in MALDI-TOF, antimicrobial susceptibility is not possible. Viruses cannot be identified by this technology, and it identifies only microorganisms which are present in its database. And so if you isolate something unique and something new, this equipment will not be able to identify them. And in our country, most important thing is equipment cost. The laboratory will not, in the small clinical laboratory will not invest 2 crore rupees for only identification of these organisms. Then we come to immunological and serological methods. That means for detection of antigen of microorganisms. And if we are unable to detect the antigen, we detect the antibodies. So older immunological methods were precipitation, agglutination, and complement fixation. They are virtually outdated except for few agglutination tests like Vidal test that we use, or TPHA test for syphilis. We hardly use any for these older immunological techniques. So we nowadays use the immunoassays, that is enzyme immunoassay, immunofluorescence, and radioimmunoassay. Again, we cannot do, for the restriction of the time, we cannot go into the details of all these technologies. See, I have already told you that each technology needs one separate class for that. So again, what is ELISA? It is enzyme-linked immunosorbent assay. We have both qualitative and quantitative ELISA techniques. Commonest method used is the sandwich method. Here by using the ELISA, we can detect antigen as well as the antibodies. These are some examples of how the principles of the ELISA. Again, we will not go into the details of all these things. The advantages of ELISA is that it is very rapid. It is very simple technique, fairly economical, and visual reading can be done. Color change. If you get the color change, you can give the idea whether it is positive or negative. Disadvantage, it can give you the false positive results. It may not be able to differentiate the present infection from the past infection, because in some cases, once the antibody come in the body, they will remain in the body for years together. And some of the organisms, the antibodies will remain lifelong. So if your antibodies are positive, you cannot say that whether it is a fresh infection or whether it is an old infection. And then some basic training is required for performing these tests. Then there is a modification of ELISA, which is called as the Western blot technique. This is basically two-step. We can call it as a two-step ELISA. In the first step, we do electrophoresis of the antigen, mixed antigen or the microorganism. So different antigens, they get separated according to the charge and the size, which we can see here. And then we do the ELISA, and different antibodies which are present there, they will get attached to the respective antigens, and you get a different bands. And on the basis of the presence of different bands and different positions, you identified the microorganisms like this. So this is useful, and this is very commonly used in the diagnosis of the HIV infection, HIV-1 and HIV-2, because there is a lot of cross reaction between HIV viruses and some other retroviruses. So some of the antigens, they are similar to each other, and therefore we have to try to detect more number of antigens to increase the specificity of these tests. Then again, same immunofluorescence. In ELISA, we use the enzyme dyes, and in immunofluorescence, we use the fluorescent dyes. So what you require is the, in ELISA, you can have the visual reading, or you can have a spectrophotometer where you can see the color intensity. Whereas in fluorescent microscope, what you want to see is the fluorescence emitted by the fluorescent dye, which is attached to the antibody or to the antigen, whichever we use in the immunofluorescence technique. The biggest disadvantage of immunofluorescence is the false positivity. Because what happens that these fluorescent dyes, they have got a natural affinity for certain proteins, and especially the immunoglobulins. So they may give you lot of false positivity in presence of, especially in presence of the antibodies. So that is the disadvantage. Otherwise, it is highly sensitive, very rapid, and almost comparable to ELISA and the radioimmunoassay. Now, we don't talk much about radioimmunoassay because the radioactivity is banned in most of the places. And you need a special equipment for measuring the radioactivity. We cannot measure the radioactivity by our naked eyes or in the fluorescent microscope. Then these serology techniques, they can be done manually, or nowadays automated machines are available. They will do everything. You just have to put the sample in the machines, and these machines will load the sample in the plates. They will mix the reagents, they will incubate, and they will give you the results also. So a large number of small and large automated systems are again available for serology. Basic principle remains the same. Then comes the molecular diagnostics. Now, again, molecular diagnostic is basically the direct diagnostic method. In this, what we detect in bacteriology, we show the presence of microorganism or we grow the microorganism. In indirect methods, we show the presence of microbial antigen or like its thumbprint, microbial antibody against the microbe. So for example, if you are able to detect antibody against Salmonella typhi, you will say that the patient is suffering from typhoid because his body has produced the antibodies. But in molecular diagnostics, we again come back to the direct diagnostic method where we directly detect the nucleic acids or the genes of microorganism. So in molecular diagnostic, we detect the genes, specific genes for specific microorganisms. So what we do, we take the specimen, pus, sputum, blood, anything you can take. You take out the nucleic acid. These nucleic acid, they are present in the nucleus of the cell. So what you do, we do the lysis of the cells, and the DNA or RNA of the microorganism is extracted. It comes out of the cells. And then we take the DNA and RNA and that we amplify. Amplify means we prepare the copies of the gene which we require. So multiple copies are prepared and then we detect the presence of that gene. So this is basically the principle of the test. Very quickly, I will let you know. See, you people belong to the pathology department. So most of the things you know. But for the benefit of postgraduates, I am trying to tell you some basic things also simultaneously. So this will be just a sort of revision. See, what we do, for amplification, we use the technology called as polymerase chain reaction, and everybody knows nowadays something which is called as PCR, है ना? कोविड के वजह से सभी को मालूम है, right? Now what we exactly do in PCR, we extract the nucleic acid, DNA or RNA is extracted. So this is for example, this is the DNA which has come out of the specimen. I will show you what happens to one DNA. We increase the temperature for about 1 minute up to 94 degrees centigrade, and this double helical structure, it gets separated from each other. This is what we call as the denaturation of the DNA. Then what happens? We put certain primers. These are the small oligonucleotide sequences of the genes which we want to amplify. See, this DNA is very big DNA. So if we want to amplify only this much portion of the DNA, जितना इसमें picture में दिखाया है. So what we do, we take one primer which moves from here to here, another primer which moves from here to here. So these two strands are separated. One primer will get attached here, another primer will get attached here, a third primer will get attached here, and fourth primer will get attached here. So what happens that we decrease the temperature from, we put the mixture here, and what we add here, we add phosphates, sugars, purines, and pyrimidines, because we want these strands to grow, these primers to grow from here to here and from here to here, right? So what we do, we will need, see, DNA का structure तो आपको मालूम है, phosphates है, deoxyribose sugar है, and purines and pyrimidines है, and the adenine, thymine, cytosine, and guanine. So we add all these in the mixture and then decrease the temperature to 54 degrees centigrade for 45 seconds. So these primers, they will get attached here. And then what happens in the third step, we again increase the temperature to 72 degrees centigrade for 2 minutes. So what will happen? This primer, it will start growing like this, and this primer, it will start growing like this. Now, what happens that if this primer has to start growing like this, it only by change of the temperature it does not happen. It will need some catalyst or some trigger to grow, है ना? उसको बोलना पड़ेगा ना, चलो grow होना चालू हो जा. उसके लिए you need an enzyme which is called as the polymerase enzyme. So in this mixture we add polymerase enzyme also. So this polymerase enzyme, it will trigger the growth, and then this primer will extend because you have put deoxyribose sugars, you have put phosphates, and you have put purines and pyrimidines in this mixture. So what will happen in about 1 minute plus 2 minutes, 3 minutes plus 45 seconds, 3 minutes, 45 seconds, approximately 4 minutes, one DNA molecule, it will become two DNA molecules. एक ये बन जाएगा और एक ये बन जाएगा. So one DNA becomes two, then two will become four, and this is one cycle. So at the end of second cycle, they will become 4, 8, 16. So they will increase in the geometrical progression, and in 1 hour, you get 2 million copies of the DNA. 2 million यानी कितने? 20 लाख copies आपको मिल जाती है, and then you can identify which organism is there. Now detection is done by the electrophoresis. This is a ladder, and here we put these amplified products. The जो भी amplify हो गए ना, so इतने million हो गए. So we put them here and do the electrophoresis, and depending upon the size of the gene, you will get different bands here in the electrophoresis, and depending upon the presence of different band, we can identify different genes which are present here. So like this, we make the diagnosis by the PCR. Now, what are the steps and what we have to do in PCR? Collect the sample. This is the example of the nasopharyngeal viruses. Because of COVID, I have put this sample in the proper container, transport it to the laboratory. In the laboratory, you receive the sample. Then what we do in the laboratory? Extract DNA or RNA. Why RNA? Because there are some viruses which do not have DNA, which are only RNA viruses. So in such cases, we extract the RNA, and here one step is increased. We do RT-PCR. RT means in the first step, we convert that RNA into DNA, and then we amplify the DNA. Again, there are so many variations and so many different things are there in PCRs. So PCR training requires a whole, at least 2 to 3 days, let me tell you. If we take only theory, it will require the whole day. And if you want to show you different techniques, it will require 2 to 3 days. I am telling you only the important salient features of this technology. Now, here we do DNA and RNA extraction. Then we put everything in these wells and then put them for amplification. And after amplification, electrophoresis, we do the reporting. So these are the steps which are involved in the PCR, and this happens very fast. In 2 hours to 4 hours, 5 hours, we can give the report. Now that first thing which I have told you is a conventional PCR. Now here people have come up with the real-time PCR, and it is one of the most useful diagnostic technology that we have nowadays. One is the qualitative PCR and another one is the quantitative PCR. So all real-time PCR systems, they rely upon the detection and quantization of the fluorescent reporter, the signal of which increases in direct proportion to the amount of PCR product in the reaction. इसमें क्या होता है? There are fluorescent dyes which are put in the reaction mixture. And what happens as the double stranded DNA starts forming in your PCR, with every cycle, the number gets increasing. These fluorescent dyes, they start binding with this double stranded DNA. And as number of the DNA increases, the fluorescence increases. The machine reads the increase in the fluorescence and it will produce a graph. And this graph is interpreted by the machine itself, and it will give you the report whether it is positive or not, like this. What happens that here the fluorescence, the number of DNA molecule, they are increasing. This is the cycle, 1 cycle, 2 cycle, 3 cycle, 4 cycle, like 30, 40 cycles. And this is the amount of the fluorescence which is being generated. So what happens initially, there will be less number of fluorescent dyes, and then as the number starts increasing, this graph, it climbs up, and you get the such type of graph for different types of the genes which are available here. And like this, we identify and make the diagnosis. Now, it is interesting that in the previous PCR, we get different bands for multiple genes. Here you get different colored, beautifully colored graph which goes up for different genes. And it is very easy to interpret in such cases because there are different fluorescent dyes used for different genes. So you get such type of result. You see, these are the cycles. So this graph, after 18 cycle, this 20 cycle, 22 cycle, this graph has raised after about 28 cycles, right? And these results are given on this side also. Now, in COVID, just for your curiosity, I am telling you that in COVID, people used to ask, what is the CT value? What is the CT value, right? So it is nothing but the cycle threshold. Cycle threshold means the cycle in which the graph has started rising. So if the number of viruses are more, the threshold will be less because the graph will rise earlier, right? So lesser the cycle threshold, more is the viral load, more is the cycle threshold, lesser is the viral load. That you understand, right? So now this graph, this is the cycle threshold for this particular is 20, for this is 22, like this. Okay. Now, so why real-time PCR is very popular? It has got a very low turnaround time, 1 hour to 2 hour hardly. And what is the advantage? There is a simultaneous amplification, detection, as well as the data analysis. So within 2 hours, you get all the results. See, in conventional PCR, what we have to do? We separate everything, then we put it in the machine. There is amplification. You take it out, then you put it in the for electrophoresis, and after electrophoresis, you put it in the gel document system, that is transilluminator, and then see for the bands. Now here machine does everything, and during amplification itself, it starts detecting, and at that same time, it starts the data analysis. So this is a big advantage. Secondly, it is a closed system. So what happens? You just have to add the specimen to the machine. And once you add the specimen to the machine does everything. So there is a less chance of contamination, less chance of human error. So no additions are made after the specimen is added, and there is a control of the contamination. Third is numerical results are there as well as visual readouts are there. So there is a less subjectivity and you are able to monitor the amplification efficiency of the machines. So these are the distinct advantages of the real-time PCR, and that's why it has become very, very popular nowadays, especially for those laboratories who are doing the molecular diagnostics. Now the question is molecular test advantageous over routine microscopy or culture? Yes, it definitely has a great advantage. What is the first advantage? It is highly sensitive. See, theoretically, it can detect the presence of even a single organism in your specimen. Of course, in practice, it can detect about 100 organisms in your specimen. Secondly, it has got a very high specificity, because if your gene is very specific, then your technology is equally specific. So it can detect specific genotypes. It can determine the drug resistance also, and it can predict the virulence also, because we can detect the genes which are responsible for the virulence of microorganisms also. It has got a very high speed because it is definitely quicker than the traditional culturing of all the organisms, not certain organisms. Plus, it is quicker than the conventional PCR also. The assays are now automated and it detects the pathogen, directly the pathogen and not the immune response. So we can very certainly say that this is the current or the fresh infection. So what are the disadvantages? So I have told you that every technology has advantages and disadvantages. So the first disadvantage, as on date, it is expensive. But you understand that if you keep on doing a large number of tests, then the cost comes down gradually. The cost of machine, investment cost of the machine, and running cost as well. Because if you remember that during the COVID pandemic, when people started doing the PCRs, the cost started from 4,500. People have even charged 6,000 rupees per test. And then it came down to hardly 600 rupees, 300 rupees, 200 rupees, like that. So the costs had come down tremendously. So if we keep on using these molecular tests, this will not be the problem in future. But as on date, it is definitely expensive as compared to the other techniques that we use. Then we will miss, of course, we will miss new organisms, because whatever the genes we know, we will be able to test only for those genes. So if there is an unknown organism or the new organism, we will definitely going to miss, which we do not miss on the conventional culture technique. And there may be a problem with mixed cultures, especially if the organisms are similar, then again you may get some false positive or false negative results in molecular diagnostics also. Now, what happens that these these have been automated, and for tuberculosis, we have got two beautiful technologies. One is GeneXpert and second is TrueNat. The difference between GeneXpert and the TrueNat is that this is a point of care test, POC test, and this is a semi point of care test. Now, this TrueNat is manufactured in India. It is approved by WHO. It is approved by RNTCP, National Tuberculosis Control Program also. GeneXpert is also approved by both. This technology is equally sensitive and specific as that of the GeneXpert, and therefore the advantages of this TrueNat is that it is the reagents and service is easily available as compared to GeneXpert, and definitely since everything is manufactured in our country, the cost comes down substantially. So as compared to GeneXpert, this technology is very economical, but for purchasing in the laboratories also, as well as for the patients also, because running cost is also very low. Now, these are the two technologies, and nowadays these technologies, they detect COVID virus also, swine flu also, dengue also, malaria also. See, so these technologies, they have got added benefit nowadays, and these technologies, they are nothing but fully automated real-time PCR systems, and people call them as CB-NAT, cartridge-based nucleic acid amplification. So whenever any PCR technology is used, it is called as nucleic acid amplification test, because it is the nucleic acid which we are amplifying. We are preparing the copies. So you call them as either PCR or you call them as nucleic acid amplification technology. Now what happens that if you are suspecting COVID virus, you will advise for PCR for COVID. Then you think that no, COVID is negative, so it could be a swine flu. So you will advise for swine flu, right? Then you will say no, it is not even swine flu, it could be influenza. So you will advise for influenza. So for a diagnosis of the viral sore throat or the rhinitis, if you have 4 or 5 organisms, you will advise 4 or 5 different tests. And what laboratory will do? You have got one sample, 4 tests are advised. You perform 4 different tests. So result takes hours to days. Then you will get 4 individual results, and then the patient stay is longer. You are unable to make diagnosis. Again, everything is delayed. So morbidity is increased, everything is delayed. So can we have something where you have one sample and do do multiple tests simultaneously? For example, if somebody is having throat infection or somebody is having pneumonia, do you have a test where if you have 20, 22, 24 organisms which will commonly cause pneumonia, if you can test them in one sample in a single go, will it not be an advantage? The scientists call it as a syndromic approach, but it is a very simple thing that whatever microorganisms they we are able to detect the group of organisms. You see, in culture, what happens? You inoculate it on the plate. If there are 10 different type of bacteria which will cause a certain infection, the organism which is causing the infection will grow on that medium. So you don't have to perform 10 tests, but for molecular things, you have to do 10 tests because you will be identifying different genes. So what they have done here is that what is called as biofire or full film array systems. Now here what they do, they take one sample. There is one chip on which there are so many holes, spots. You can call them as spots. And these spots are for different genes of different organisms. And then and they will put the sample here, and whole of the real-time PCR, it will occur automatically in the machine on this particular chip, and it will give you multiple results in one sample. And then immediately within 1 hour, you get the results. Now, what are the different panels that are available? One is the respiratory panel. So all respiratory pathogens, all organisms, common organisms, I'm talking only about common organisms, which will cause the septicemia, common organisms causing gastrointestinal panel, meningitis, encephalitis panel, and the pneumonia panel. Now, we have to understand that in culture, we are able to grow only bacteria. But in this technology, molecular technology, in the same panel, we can detect bacteria, we can detect viruses, we can detect fungi, and some parasites also, especially the protozoan. So these have got a great promise. So like this, you can see here that this is one panel which is causing, this is the diarrhea panel, or this is the, I think this is the respiratory panel. And you can see that here it is going to, it is showing this Escherichia coli positive, Moraxella positive, and Staphylococcus aureus positive. Now these reporting, and you can see here it is semi-quantitative also. So number of bacteria, they are showing it 10 to the power 4, 5, 6, 7. So here most of the bacteria, they are Moraxella catarrhalis, which is an opportunistic pathogen. Very few Staphylococcus aureus and intermediate Escherichia coli. So like this, you get the final report. See, this is showing this so many organisms, they are detected in this biofire. They can be detected in this biofire, and in this test, what they have detected: coronavirus OC43, human metapneumovirus, human rhinovirus, influenza B, and parainfluenza virus 3. Now see, in a way, this is an advantage, but in a way, this is a disadvantage also, because there are many viruses which have just colonized your respiratory tract, and they also are detected here because they are just present there. So it will be ultimately your choice and the clinician's choice to decide that which organism is causing the infection. So let us see the advantages. It has got a biofire or the film array. It has got a less time to targeted treatment, shorter antibiotic durations. It significantly reduces hospital length of stay. It avoids unnecessary use of the antibiotics. It promotes antimicrobial stewardship group and more informed infection control decisions. These are for the hospitals, right? What are the drawbacks? See, there are problems that the amplification may not be proper because there is self-inhibition among different sets of primers. So there is a low amplification efficiency. So the sensitivity drops down a bit. No identical efficacy or different template. Now what happens if you have different templates from different companies, you get the variation in the results, right? And single sample throughput is not there, and the cost per test definitely is very huge. You will the laboratory will be spending somewhere between 10,000 to 15,000 rupees per test, and therefore the patient cost will also increase further. Then comes the gene sequencing. Gene sequencing. This is the latest technology which is available. Now, what is gene sequencing? It is basically the process of determining the sequence of nucleotides within a DNA molecule. तो उसका पूरा आपको पूरा बायोडाटा ही निकल जाता है ना? Genetic biodata for organism. So it is looking to sequence and map of all the genes which are present in the microorganism. So it means that you are reading the nature's complete genetic blueprint. So you get the correct blueprint of that organism, and if you know the blueprint, you can identify the organism very easily. Now, gene sequencing is divided in different generations. So there was a first generation gene sequencing, and all other second, third, and fourth generation sequencing, they are called as next generation sequencing. So people will talk to you about NGS. आपने को लगता है what is NGS? NGS is nothing but a next generation sequencing of the genes. Again, we will not go into the details because this is in itself is a separate topic. But first understand that the first generation, that the first sequencing in the world for identification of the genes was done by Sanger. And this Sanger sequencing is done, and that fellow, he has done it manually. So it must have been so difficult, let me tell you. But nowadays, fully automated systems are available and people have resorted to the sequencing. Now, the difference between the first generation and the next generation sequencing is that in first generation sequencing, very small and tiny sequences, they can be studied. Here, there is simultaneously, you can study hundreds of different genes, and the profile, it comes out very fast. Now, sequencing is done by either by hybridization or by synthesis. Again, we will not go into the details of all these things now because it is a very, very new technique, and we are not going to utilize this technique at present immediately, except in the big institutions. And secondly, the technology, the machines, they are fully automated machines. We only have to understand the principle behind them. Again, these are the first and second generation technologies, and different companies have prepared different platforms. And these platforms, they are available nowadays. Commercially also, they are available. And each test will cost you around 18,000 to 20,000 rupees. Now, what happens that a sequence of a microorganism, it will create a plethora of data. I'm not telling you that it is large data. It is huge, huge, huge data. And it will give you the plethora of data, and you don't know how to interpret and how to go through this data manually. It is virtually impossible because each organism, although it has, my bacteria have got only one chromosome, but there will be so many genes, let me tell you. So what happens, there is a branch called as bioinformatics. They have got the classified information about different organisms and different sequences and different mutations that occur in these sequences. And we can take help of this bioinformatics. The sequencing that you have got, it is sent to the bioinformatic library, where they have got the stored information for huge number of sequences. So what they will do, they will identify, they will analyze your sequence, and they will submit you the report. So there is a there are multiple steps basically. कि उसमें डाल दिया और होगा ऐसा नहीं रहता. There are basic steps. There is a primary analysis, then secondary analysis, final review takes place, then reporting is done. And if you identify the variant, the new organism or new genetic pattern is identified, then you have to do the follow-up also. So like this, these systems are nowadays, they have been working. So what are the advantages of next generation sequencing? Next generation sequencing may be used for whole genome sequencing. We call it as the WGS. on bacterial isolates from a single patient or from multiple patients. It offers rapid bacterial identification and has the capacity to differentiate between different clones. And secondly, the technique is becoming increasingly automated and the cost continues to decrease. For example, now Mycobacterium tuberculosis, if you try to grow it, it will take, even in the automated machines, it will take 15 days to 20 days. Then susceptibility testing will require next 15 to 20 days. But if you do the whole genomic sequencing, so immediately within a day, you will get the information that yes, this is Mycobacterium tuberculosis and it is sensitive to such and such anti-tubercular drugs and resistant to such and such anti-tubercular drugs. So this technology is very rapid, very sensitive, very specific, and very useful. But at present, technology is not available everywhere. And secondly, it is very expensive. So what are the limitations? Next generation sequencing generates a complex data that needs interpretation by clinical microbiologists. Sensitivity and specificity of individual platforms cannot be compared directly. Next generation sequencing cannot be used to detect RNA viruses without modifying their protocol, because you will have to create a DNA copy of that RNA virus by reverse transcriptase PCR and then identify. Then it is unclear about the antibiotic resistance, because what happens, many times genes are present but they don't manifest. So phenotypically it is not showing any resistance, so it will be killed by that antibiotic although the gene is present. And again, in our country, cost is the important. So you don't automate to get rid of human beings, you automate to amplify the human beings. And that's why so much of amplification, so much of automation has come in the microbiology. We have to understand that skilled expertise of microbiologists and technical staff are still indispensable. Automation can only be of immense contribution if it is properly followed. And automation is the buzzword, and therefore we are going in for the automation, and it is the future technology. Now, ladies and gentlemen, we have to understand that although so much of automation has occurred in microbiology, the basic microbiology technology, the basic techniques of smear examination, culture, antimicrobial susceptibility testing, the detection by ELISA, immunofluorescence, they are still used everywhere throughout the world, and not only in our country, but throughout the world. We are not backward and lesser than any other developed country. We have got all these automations in our country, in many of them in our clinical laboratories also. So we need to understand these technologies. We need to make good use of these technologies. What we do in our clinical laboratory, we mix the manual technique with the automated technique, and we give the better results at a very affordable cost to the patient. That is what that's the system that is followed by in our country. So automation is the future, and we have to adopt. Thank you very much for your patient listening, and I am sure I have not exceeded much the time. I have finished in less than 1 hour. So if you have some questions, you can ask me. If possible, I will answer them now, or otherwise, if you write the question and send it to me on WhatsApp or on mail also, I'll try to answer your questions. Thank you very much.
Thank you. Thank you so much, sir, for your throughout thought-provoking lecture, which has added depth of our understanding. Thank you so much, sir. We appreciate the knowledge you have imparted. Now, I request Dr. Abhinandan Mukesh, sir, chairperson of today's webinar, to add valuable comments, which will undoubtedly contribute a lot. So, Mukesh, sir, please.
Thank you, madam. Good afternoon. MCM approved Naidanika 2024 webinar is organized by Rognidan Vikriti Vignan PG Association. Among 4 speakers of webinar, today Dr. Yagnesh Thakar sir has opened the webinar with his informative lecture. I congratulate and thank Dr. Thakar sir for his lecture, which was in a simple way for such a difficult topic like microbiology, and also he has finished it in a time of 60 minutes, and shared a lot of things like only 100,000, only 10,000 organisms per ml can be seen microscopically. Such things are very much useful for the students to understand the automation needed in the field of microbiology. And explain automation and current advances in microbiology very nicely, like information about advanced machines like multi-top, also explain limitations and cost of the automation, and automation cannot able to identify organisms. We have clearly mentioned some limitations of the machines also. He also explained about ELISA, which is useful for antigen and antibody detection. And but he also mentioned the limitations of ELISA, that it cannot differentiate from past infection from present infection, which has to be considered by all clinicians and practitioners. Again, he explained about Western blot, that is very commonly used test, and he has nicely explained. Then he told about the PCR, uh, where the DNA or RNA is extracted, and the advanced, that is real-time PCR, how it is useful, sir has explained very nicely. Then fluorescence dyes, which are helpful for detection in real-time PCR, we explained in detail. Sir has nicely explained the concept, that is cycle threshold is inversely proportional to the viral load, which is very important as to be noted by practitioners while understanding the reports. Whereas toward the limitations of traditional testing, but not only sharing the limitations of traditional testing, there has shared the biofire film array systems to overcome the limitations of traditional testings, and which can detect bacteria, viruses, fungi in the same test, which was very fantastic information about biofire film array system, which is very new to all also. Then Sir has explained nicely about gene sequencing, that is NGS. He told the advantages of that is next generation sequencing of gene. Then Sir has nicely explained about bioinformatics use, which is now good trend in the gene sequencing. Sir has explained the microbiology advances in a short time. I really thank Sir for sharing time and sharing his thoughts. We came to know his experience and knowledge from the lecture. I thank Dr. Yogi Sakar sir for his informative lecture. And I thank first to Dr. Waghe sir for inviting such an experienced and knowledgeable speaker for today's lecture. I thank webinar coordinator Dr. Amle madam, Dr. Anna Abrashik madam, then Hemant sir for taking efforts to organize the webinar. I'm confident that all the 84 delegates who have attended the lecture have benefited a lot from the Sakar sir's lecture. I thank all delegates for attending the webinar. I thank Dr. Chauhan sir, Dr. Gaonkar madam, Shirke sir, Vyavare sir, Sachin Deva sir, Archana Kulkuri madam, who are attending the webinar. So I thank Waghe sir once again for arranging the lecture. Sir is known for arranging such programs. He is having the vision. He connects the knowledgeable people with positive approach and make benefit to the students, PG students, faculties, and researchers of medical fraternity. Thank you. Thank you, Thakar sir, Amber madam, Vake sir.
Thank you so much, sir. I request all delegates to put their queries in chat box, or you can directly communicate to the sir. Now, I request Dr. Manisha Talekar ma'am, Medical Officer, CCRS, Nagpur, to deliver the vote of thanks. Towards you, ma'am.
Thank you, ma'am. Respected speaker, Dr.
Madam, you are not audible. Please unmute yourself.
Her net is interrupted, it seems. So somebody will have to call her.
Till that, I request Dr. Subhash Wage sir, President and and Organizing Secretary, RVPG Association, to conclude this. So I think ma'am is online again.
Yes,
Ma'am,
ma'am.
please continue with vote of thanks.
Thank you, ma'am. Our respected eminent speaker, Dr. Yagnesh Thakur, sir, Organizing Secretary of RVPJ, Dr. Subhash Wage, sir, and all the dignitaries, a very good afternoon to all of you. It's a privilege and a great joy for me to extend the vote of thanks who have contributed in this webinar, Nedaniki 2024, organized by RVPJ. At the
Ma'am, you are not audible again.
I think her network is disrupting, and on behalf of RVPGA organization, let me first offer my very deepest gratitude and to my teacher, Dr. Yagnesh Thakur sir, for his sparing his valuable knowledge and valuable time for our peoples. Madam, I will continue now. And
yes, sir.
I think your network is breaking again and again. So I take this opportunity to thank everyone. The chairperson, our today's chairperson, Abhinandan Muke sir, Dr. Dipali Amle madam, today's coordinator, and all the participants who have joined this academic feast, and Dr. Manisha Tadekar madam. I thank everyone, and I hope that all the participants, they have sufficiently sensitized for our upcoming uh, Pathcon hands-on training workshop on microbiological diagnostic methods. Sir has provided us a very beautiful and in a very lucid manner the insight to the technological advances and advanced technologies in the field of microbiology. And if you even though the huge advances are taking place, but he has also stressed out the importance of traditional methods which we are doing in the microbiology, in the field of microbiology. You see, in Ayurvedic Vikriti Vignan PG course, there is a fourth paper which is separately on the microbiology. So, so in this context, I will request every participant to join us in a Pathcon hands-on training program, where there will be a beautiful demonstration of all these technologies practically, and sir will be the hands-on training program will be presided over by the Thakar sir and Uge Muge madam and his entire team. Sir has a beautiful setup at Vishakha Labs and the latest machineries, and hopefully we all will get benefited from that and I'm expecting you all and inviting you all to join the Pathocon 2024 hands-on training workshop on microbiological diagnostic techniques. Thank you. We can conclude the meeting now. Thank you very much again, sir. Thakar, sir, thank you very much.
Thank you so much, sir. Thank you, sir. Thank you, ma'am.
Thank you, ma'am.
So I will leave the meeting.
Yeah, okay.
Yeah.